And so the Soredia are just like the packet of fungal hyphae that's usually wrapped around nicely the algae or the cyanobacteria, and then that breaks free as it gets disturbed, as the lichen gets disturbed with weather or an animal. And so that's a good way to disperse in natural settings. The idea that, excuse me, I have down in the notes too, if you want to read a little bit more about it, but there's been some debate about lichens that, is it always a win-win symbiotic relationship, or can it sometimes be where the fungus is kind of trapping the algae and cyanobacteria and taking advantage, kind of like having the algae or cyanobacteria as kind of like a hostage situation. Because they're so well, the fungal hyphae is so well wrapped around the algae or cyanobacteria, they sometimes wonder about that. And then in the lab, when you try to grow these fungi separately, they can grow, but they're pretty weak and weak growth, like slow, sparse growth. Whereas on the other hand, the algae and the cyanobacteria grow quite nicely, but that could just be because we don't understand the fungi well enough. So you take that with a grain of salt. Just be aware that there's some debate that sometimes they are thinking, well, maybe it's not always just a win-win relationship. Maybe the fungi is taking advantage. Yes.
Does this algae do photosynthesis for
Yes. Yes.
a power source that the
So yeah, the algae and cyanobacteria are great for the fungus because they do photosynthesis and share the sugar. So it's great, but on the other hand, the fungus seems to provide a lot of minerals because they're so great at excreting out enzymes to break down rock and other material. So yeah, it's an interesting dynamic, and there's still a lot of mystery when it comes to lichens. So one thing they do know for sure is that they do grow very slowly. So if you are interested in lichens and want to have some around you in your home, or if you have a terrarium or something, just be aware that you need to be careful how much you take. I went on a summer workshop. Dr. Robertus and I went, this was years ago, here in the UP, Michigan MSU lichenologist came up. I love that word, lichenologist. And he was like at the time, one of the top lichenologists in the country. He was well renowned. Of course, I'm blanking on his name, sorry, but he was really famous. Anyways, he came up to give the workshop, and it was so interesting. We got to go out and take samples, but when we did go out, because there's like 24 of us, he's like, be careful where you step, don't take too much of the material. But so we took, we were very respectful and took a little bit and then came back to the lab, and excuse me, we're able to examine them, macro and microscopically. But yeah, it was a lot of fun. We learned a lot from him. He's retired now, but they do grow very slowly, and often they look dead if you didn't know any better, because they look dry and brittle and just dehydrated. But that's their version of suspended animation, so to speak. They're just really good at going dormant. And if you have some extra water in your water bottle that you wanted to share, you can always water the lichen, and you can sit there if you have a half hour or so. You'll see it start to green up. The algae and or cyanobacteria will start to waken up, so to speak. It'll start doing photosynthesis. You'll start to see the green chlorophyll if you look closely. So it's a really nice strategy. It lets them, if you think about all the lichens that you see when you hike around here that are just on rock, exposed to the bright sun, it's an existence that's challenging. And so sometimes they have to do some drastic measures in order to stay alive through the long haul. And so they're very, very good at handling extreme environments. In fact, people who have looked for them in all the different continents find them. I mean, even, in Antarctica, you can find them. They're a little bit more sheltered there. I'll show you a picture of that. But all the way from, Arctic, Antarctica, desert conditions, they're everywhere, rooftops, all kinds of concrete and artificial surfaces. So they are well known for that ability. And because part of the partnership is photosynthetic, they are essentially primary colonizers. They're part of the base of the food web, so to speak, because they can, and then they can often get established in environments that are very, like if you can imagine a fire wiping through an area and, just kills everything in its path. Lichens could be the first in, because they're just capable of landing on some rock material and they'll start growing. And so they help too with over a long time period. They can help create soil because of the fungi, especially ability to break down rock material and complex minerals, aggregated minerals. And so they're well known for that. The other nice thing that the cyanobacteria can do, so Nostoc is the cyanobacteria partner. So it's a bacteria, and it has the extra ability, not only can it do photosynthesis, but it can fix nitrogen. And that is huge. So that's another reason why they're great primary colonizers, because they can enrich that habitat with nitrogen if it's nitrogen poor to begin with. And so certain bacteria have the ability to access dinitrogen gas in the atmosphere and convert that to ammonia, which is the source then that living or other living organisms can work with. So there's only a handful of bacteria that can do this, and they're incredibly important for nitrogen cycling on our planet. So if you think about nitrogen poor soils, like believe it or not, tropical rainforests often can be defined that way. The soil systems can be poor in nitrogen. They out in the Pacific Northwest habitats, they can see that it really does help. And then of course, more extreme like desert and tundra areas, having that partner that can fix nitrogen is definitely, if I was a fungus, I definitely look for that kind of partner in these types of habitats. So here you're seeing pictures of cliff faces in Antarctica, and they zoomed in on some of the cliff faces on figure B there. And you can actually see how the lichen, believe it or not, that's lichen that you're looking at. And, they did have to study this pretty carefully, but you're seeing all that. I think you can see from even in this lighting, the greenish area there, and then the white and black. That's the black is the fungus, and the greenish, whitish area are the algae. So, hiding away in the rock, that's pretty sneaky. So they can exist in Antarctica long term by going, I think they use the word here, endolithic. I don't know if they use it in this one, but that's the fancy word. You don't need to know that, but they will say, oh, that's an endolithic algae or lichen. That means it's in the rock. Litho refers to rock, endo within. So that's pretty extreme. So this is a classic set of data showing how lichens are sensitive to air pollution. So this was a study done over in England many, many years ago. But it's just a classic representation of the idea that if there's a lot of air pollution in particular, lichens tend not to be found in high densities. Now, you've had a chance to walk around Marquette and the surrounding areas. Do you think you see a lot of lichens? That makes me happy. That makes me very reassured that our air quality is pretty good. And so you can find, it's really funny after Dr. Robertson did that, when we did that workshop, it was so Friday, Saturday, Sunday, intense, all day long, 3 days in a row of just lichens. And then after that, I could see lichens everywhere. It was like all of a sudden my eyes were open and you could just see and it lingered. Like I can still do that. So I encourage you to really kind of zoom in on them for a while, and then when you start looking, you'll see them everywhere, especially around here. Thankfully, we do see them right in town, right downtown. I can see them if I'm walking downtown, and so they don't really have a way to get rid of pollutants. They don't have an excretory system like we do. So they tend to accumulate. And one of the first things that tends to get hit is chlorophyll. And obviously, if you're doing photosynthesis and you don't have good chlorophyll, not a good thing. And so you can get all the way to death of a lichen or just severely impeded in their growth. Sulfur dioxide, that air pollutant seems to be one of the strongest impacts, on their ability to survive. So this data set that you're looking at is the 0 mark on the graph is the middle of the city in England, Newcastle upon Tyne in England. And you can see it's not until you get beyond 10 kilometers out from the center of the city that you finally see lichens popping up. So wouldn't want to live there. Hopefully after now, this was a couple decades ago, hopefully by now it's better, but I don't know. Then a student in BI 310 did this project, Kathleen, this was a few years ago. And so she did a really nice study where she was kind of asking the same question as what they were doing over in England. And she was able to show, she took some nice sampling from Marquette's, city center essentially, and then went out in different directions. And she was looking for the presence of lichen. I think she also was looking at if she did find lichens, like, I think she focused mainly on tree trunks. And if she did find lichens, were they on like the north facing tree trunk side, the south facing? So she did some really nice studies and presented that in the poster session for BI 10. And you can see that you do see this trend, right? Of there's more lichens as you get further from Marquette, but we still have good coverage in Marquette. So not like the other data set I just showed you where there's nothing, they couldn't find any. And so, so north and south side of all the trees sampled. And so where you see the asterisk, those are all significantly more growth compared to Marquette, but still, you still have some nice data showing that there's plenty of lichens. So the lichens you'll see in lab, hopefully tomorrow, we tend to talk about what they look like and group them that way. So by the way, I don't think I introduced the term thallus. So when you look at a lichen, often they'll call the whole thing the thallus. I usually say body, the body of the lichen. It means the same thing. It don't get too hung up on that, but you might see that word a lot, especially maybe on some of the slides that you look at in lab. So just be aware that people sometimes will talk about it that way. But you'll look at some crustose type lichens, which when you look at them with the naked eye, they're very flat to the substrate they're attached to. And they tend to be adhered very tightly, very low. They don't grow up. They just stick really tight and low to, we have samples that are mainly on rock or branches. And then the foliose type, they give that name because they do kind of look leaf-like. They tend to still not go very vertical. They're pretty close to the substrate that they're growing on or attached to. But they look more, they're not so crusty looking. They look more like a leaf. And then the fruticose is the third major category of thallus morphology. And those are more erect and come up off the substrate. And so that's basically the three body forms that they have grouped, and that they seem to all fit into one of those three. And so it just depends on who the fungus is and also somewhat what the algae or cyanobacteria partners are too. So this Lotharia species is definitely fruticose. It's coming up off looks like a little shrub, erect off I think this is background, maybe it looks like rock or a cliff face, maybe. Bright yellow. This one I have in the notes that it's called the wolf lichen because of their common use as poison. This is a sad story. Common use for poisons for wolves and foxes in Europe centuries ago. The lichen with its toxic vulpinic acid was mixed with ground glass and meat. Isn't that lovely? Apparently a deadly combination, you think? So the lichen was also used as a source for making a yellow colored dye. Much better story. which is not difficult to imagine. So yeah, so people who didn't like wolves or foxes in their area would do that. So hopefully that is not something well known anymore. That was, a couple centuries ago. Hopefully that's still not a thing anywhere. But you can certainly imagine the beautiful dye that you could extract from that. So that's coming back big time. I've seen a lot of fabric where they boast that it's been dyed using natural dyes from lichens. And again, I'm a little worried about that because it's like, well, lichens grow so slowly. Like, what's their source of lichen? Are they going to be unsustainable in their harvesting? So you got to think a little bit about that, but got to be careful. So this one's a little hard to tell from, especially from this angle, because you're kind of looking down on it. I could see where you might say, well, it kind of looks foliose, but technically this is fruticose. And so that's a little difficult to tell. Crustose, that's a little easier, very crusty looking. So we have a lot of that around here. I'll show you another picture of that in a minute. So these beautiful pigments that you see, if you think about it, and once you learn, that these pigments can help protect from too much sun. But, so it's like, well, yes, they need to do photosynthesis. The algae or the cyanobacteria need to do that. On the other hand, where you see most lichens, they're often on exposed rock up on the mountaintop where the sun just relentlessly beats down on them. So you can imagine that sometimes that might just be too intense. And so they will often express other pigments to help them deal with too much sunlight all at once. And so that's why they're an attractive source for dyes, is because you can get some really intense pigments being produced if you go to the right habitats. And so keep an eye out for that as you're hiking around. Some very beautiful pigments. The under the UV light, I haven't. When have you?
Pretty cool.
Does it?
They look, some of them do, they look way different than they do.
Oh, we have UV lights up in the micro lab. I should bring those down for tomorrow's lab. You just have to be careful with the UV lights, of course. Don't you look directly at the UV light, okay? And don't put your hand under it long term. I had a fellow grad student when I was in grad school. Did I tell you this story? We had a flow hood, so a flow hood creates a sterile bench space, airspace, so you can work aseptically, but it has a UV light to help sterilize that area and the bench. And he thought it was just, he was not from this country, and he had limited, he passed the English language test, but I don't think he caught everything. And so he thought it was a normal light and he left it on. He came in and he saw it was on, because often we'd leave it on overnight. And then when you come in the morning, you turn it off. He kept it on and he was working in there for like over an hour. And then like the next day he's like, yeah. And I'm like, what happened? He's just sunburned. And yeah, so he learned his lesson on that one. I'm like, no, that's a UV light, and he didn't know. I felt bad, but so anyways, don't look at the light. Don't put your hand under it. Yes, yeah, an air plant, yeah. So they're epiphytic. They tend to be that way in their natural habitat. So anyways, here's another, this is a National Geographic photo on a gravestone. So really nice color combination there. So, granite, yeah, if you've ever walked, like I like sometimes to walk 7th Street Cemetery with the ponds there and everything. And that's one source close by where I can see magnolia. There's a beautiful magnolia tree there by one of the ponds. So in spring here, eventually we'll have spring with blossoms. You should go check out the magnolia tree. It's beautiful. Magnolia trees have such cool growth forms, at least here, because they still grow low, but they have really interesting branch morphology. So anyways, foliose, this is a little bit more foliose-like, not so upright. It's still kind of flat to the surface, but I have a hard time sometimes between fruticose and foliose, unless it's like more like that wolf lichen where it's like a shrub. Sometimes it can be challenging. This is pretty nice because this is really close to the substrate. So this is more obviously a foliose, but it does kind of look leaf-like. And, all beautiful different colors. And so often too, like the British soldiers that you're seeing here, they call British soldiers. They grow upright, through those, but that's a lot to do with the way the fungal the fungal hyphae grows, and when it goes through sexual reproduction, it produces sexual spores, it'll give that growth form. And then these, that's the original color, that wasn't photoshopped, so they can be really, really beautiful. This is the one that we have at the Lower Harbor. You probably noticed all the orange, even from a distance, you can see it. So that's Xanthoria, as far as I know. I talked with Dr. Robertus about it to verify. But yeah, because lichens are so tough, NASA's interested in them. Like, yeah, yeah. So they're getting into it and just trying to see, here 18 months of space exposure, simulated Mars conditions. So they are tough organisms, but it's a really interesting dynamic how they, these different species come together and they look so unique when they form this partnership compared to what they look like when they grow separately. So it's just fascinating to see, their body when they're in this symbiosis compared to if they live alone.
Potentially. Like in Mars, I think.
Don't know. I think, they're looking for bacteria on Mars right now, and they seem to be getting, at least I'm not keeping super up to date with it, but the last few snippets I've read, they're getting more and more confident that there is microbial life there. It might be in the rocks, but you could see where maybe the lichens, you never know. Yes.
That's fine.
Right. Yeah. Yep. And then this is Ryan Rutherford. I was on his committee when he was here doing his work with lichens in the Huron Mountains, which is near Big Bay. And so he was able to do a really nice study there, and he made the new that TV show Discovering. I think he was on that show. They interviewed him.
Anybody.
I think it was him, because he's kind of the local lichen expert right now. And so they interviewed him. He had a whole like half-hour session on the TV. So I've had this question and I had to go look it up. Students said, okay, well, where, so this is fungi, algae, bacteria, did they show up before the other, like the land plants? And I'm like, oh, that's a good question. And I was assuming because it's fungus, algae, and cyanobacteria that they would have beat out the land plants, like the bryophytes that we'll talk about next. And they didn't. So I was shocked to find that lichens, they came after the vascular plants. And the vascular plants came after the bryophytes. So this is quite not where I would have put them. So I was really quite surprised about that. So, but anyways, they are a very fascinating group of organisms. So you could go on and on a whole semester. I mean, people teach like a whole class just on lichens for 16 weeks. So obviously there's a lot to know, but we got to move on. So although we're not going to move too far because now, so we've covered fungi and then fungi as lichen partners. Now we're going to talk about algae, and remember algae are part of this partnership. So we're shifting a little bit and we're going to get into algae next. So, and again, you could cover algae in take a whole semester to do it and you still wouldn't be all the way through. So you have to realize that what I'm showing you is definitely, we're scratching the surface, but for this type of intro botany course, you do need to get some exposure to algae, and in lab, tomorrow, hopefully. You'll get to experience some of that as well in multiple different sensory ways. So, okay, algae are funky. They're in the kingdom Protista. Some people say kingdom Protoctista. I tend to do that second one, Protoctista. Either way, they are all over the map. It's really hard. We have different groups of algae. There's green algae, brown algae, red algae, and there's several others. They're very complicated. And so I'm not going to go into depth, but there's certain things I think you should be aware of for this level class. Their morphology is one, okay? So what is their body type? What do they look like? So you can see when you look at all the algae out there, you can say, well, there's some that are single-celled and often microscopic in that regard. There are some that are colonial in the way they organize with each other. And I'll show you pictures of all of these. And then if they're colonial, they often have this kind of gelatinous covering or sheath that keeps them all closely together so they can remain hanging out with each other. And then there's filamentous type. There's seaweed type that look like, lettuce leaves, essentially. And then there's the kelps. So those are your options. No matter what type of algae you're looking at, they'll fall into one of these. So I do need you to know their morphology. Yes.
So seaweed is algae.
No, well, I'll show you. It can be tricky. Algae are wacky, so I'm not an expert in algae. I'll say that right out front here. I've had to do a lot of digging on my own on this. So yeah, it can get, what's an algae versus what's a bryophyte type or liverwort? You're getting into. Yes, kind of drives me nuts. Yes, yeah, it's so this whole kingdom Protista, it's like, yes, it is really, and it keeps changing as they find out more, then they keep rearranging the phylogeny, and yeah, it's a little frustrating.
It's not a plant, animal, fungus.
Then it goes into the, yep, Protista. So the kelps, I need you to know so kelps are a little bit more complicated in their morphology because you get certain parts that look different from the other parts. So I do need you to know the basics. So hold fast. We don't say roots. We say hold fast. That's what's the root-like part of it. That's what's actually helping them attach to some kind of surface of some sort. And then the stipe. So in yellow is what I want you to know. The stem part is called the stipe or stalk. Depends on who's talking. The so-called leaves are actually the blades, so no blade. And then the gas bladders are the little round structures that you're seeing here. Okay. So we don't have any kelps here in Lake Superior, unfortunately. But I've not yet done this, but I would love to scuba or snorkel with kelp forests. Has anyone had a chance to do kelp forest? I just think that'd be fascinating. And so, I mean, they rival the diversity there in kelp forests is just amazing when you think about all the other life forms there, including many animals that hang out in kelp forests. So, okay, single-celled, colonial, and filamentous. So single-celled, you tend to need a microscope, and you often see them, they're motile with flagella. And so they usually zip around. If you look at them under the microscope, they'll be hard to keep still unless you have some of that protoslow viscous fluid you can add in there to slow them way down. Colonial, you can see they're single cells, but they're all grouped together. This one's really nicely, the Euderinia. This one's a little trickier, the Volvox. That's all the same. That's not a mixture of different species. It's just they're in different phases of their life cycle. That's why they look different like that, and we're not getting into their life cycle. We are not getting into how the algae have sex, because it's all over the map. They're very creative. So, and it's just like, I give up. So we're not going to do that. And so, and then you have filamentous. Huh? Yeah, they're very inventive, so, Yeah, I tried to teach about, because that's often how we classify. We look at how they go, like with fungi, you look at how do they go through sexual reproduction, and that's how we, one major way we classify them. Are they an ascomycete, a basidiomycete? And they do that with algae too, but it gets so complicated, and they have so many different mechanisms at play. So, and by the way, when you're looking at algae in lab, a lot of the slides, I was very limited on what kind of prepared slides I could buy, what they have available. They tend to focus on how they have sex. So we're gonna ignore, like, just look at them and don't worry about, you're gonna see all this weird terminology, and don't worry about that, because we're not getting into that. So filamentous. So yeah, I don't think I'd want to swim in that lake. But now this I'd like to swim.
Oh, yeah.
Wouldn't that be just amazing? What is that? Oh my goodness. The headset thing, you have virtual reality?
Yeah.
Yeah. What's the popular one? Like, I have it at home. What is it? Oculus Quest. There we go. I gotta get back to that. I keep wanting to do that. But anyways, I keep waiting for them to come up with this. Could you, because they do an underwater one. And so I'll have it on, and I'll just be standing there in the living room. And my sons come out, what are you doing? I said, I'm at the bottom of the ocean. But they can't do a kelp forest. They do whales, like the whale will sneak up on you, and all of a sudden you'll see it in your periphery, and you're like, whoa. But kelp forest, and they haven't done that yet. So I think it's a little challenging for the programmers, but I'm waiting. It's like, come on. So beautiful kelp forests. And so this is just a close-up of the bladder, blades. Under, real situation, you see the bladders. So it's just filled with air. It's like a bladder of air, so that they can stay elevated in the water column enough to get access to sunlight. So like we were just saying just a minute ago, the phylogeny of this group changes over and over and over. This is the newest classification. Like, I didn't learn this when I was an undergrad, and a lot of this is new. So this is what I want you to know. So what I've crossed off, you don't need to know, but I want you to know, okay, so now moving forward in this class, phylum names become important. I will be asking you to memorize those now. And so phylum Chlorophyta, and who's pronouncing it, by the way, there's a lot of different ways, well, at least two. Chromophyta, Rhodophyta, or Rhodophyta, I mean, doesn't matter, I say it both ways. Dinophyta and Charophyta on the very bottom are what I'll mention. And like, they didn't have Charophyta as a separate thing, they were lumped into the green algae, the Chlorophyta. So this is a nice summary table of who falls into these different phyla. So the Chlorophyta or Chlorophyta, you can see those are the green algae. And then one way that they do classify is they look at how do they store up carbohydrate. Like when we store up carbohydrate, it's usually in the form of glycogen, but green algae tend to do starch like most plants do. But you can see the Chromophyta don't do that so much. Instead, they use oils and fats as their storage. So that's actually a criteria. So I would like you to link that to which phylum. And then chlorophylls. So chlorophylls can be broken down into A, B, C, D types. And I'd like you to note, do you see chlorophyll A anywhere? Okay, so that's important. So here in the algae, we don't see that, but we do in plants. Okay, plants have chlorophyll A and B primarily. Here you see with algae, it's B, C, and D. And then I don't care about the flagella, but that's a criteria. They do use that, but I don't want you to memorize that. That's okay. Don't worry about that. So these different chlorophylls, they're very similar chemically when you look at them, just some slight modifications. But it does create a different absorption preference of what quality of light they prefer to absorb. And so, for example, the rhodophyta or rhodophyta, the red algae, chlorophyll D is well suited if you're lower in the water column of the ocean, where it's dimmer light. Chlorophyll D is just really good at snagging the quality of light at that level in the water column. And so that's why, I mean, basically that's why you're seeing these differences. It's their habitat and how their chlorophyll evolved to adapt to absorb those qualities of light in those different habitats. Okay, so green algae is probably what you're most familiar with. Yes, so I'll start there. Mhm.
Do all algae produce like oxygen? Are there only some algae that?
Yeah, that's a good question. So when they do photosynthesis, they're oxygen producers. Yeah, they do that. And so in nostoc, the cyanobacteria, well, in lichens, they do that too. But anyways, all right, so did you have a question?
Yeah, do you want us to know what type of butterfly?
I would, yeah. Oh, wait. I was wrong. A is not on there because they're all in that group, right? There's some, that should be in here somewhere. Oh. Yeah, sorry, I misspoke. All phylum algae have chlorophyll A. I was wondering, like, what is wrong with this column? There's something weird. All right, so all of them have A, and then B, C's, and D's can also occur depending on their habitat. Sorry about that. Okay, so green algae, this one, Chlamydomonas, is like the guinea pig of the green algae. It's been heavily studied. You don't need to know its anatomy, internal anatomy like that, but they're funky. They have a huge chloroplast inside. And they use two flagella. The pyrenoid area, that's just where they do the Calvin cycle. It's like a localized spot where they can see that's where everything is located for the Calvin cycle. But that's what they look like under the microscope. They're really cute. They zing along really quickly under your field of view. So hopefully we're going to have a bunch of green algae, fresh living samples for you to look at tomorrow if we don't have a snow day. So hopefully you'll be seeing all that. You might be more aware of them causing problems, harmful algal blooms. You see that a lot in the media, unfortunately, these days. So the acronym is HAB. HABs are harmful algal blooms. So that's what you're seeing here. We have an overgrowth of algae, primarily because there's just too much nutrition, pollution essentially, which is nutrition for the algae. Get an overgrowth, and it's not just algae, it can be cyanobacteria too. But in general, especially some of the algae, depending on the species, they can excrete out some toxins that you don't want to be around. Fish don't survive it very well. So I know Michigan Tech University does have a center on their campus where I think they received some grant money along with several other universities in the Great Lakes region where they're monitoring the HABs. And so they'll put out reports, and they usually do like Google Earth imagery. Clearly I need more caffeine today. And so the amount of harmful algal blooms in Lake Superior of all the Great Lakes is less than all the other ones, but still, unfortunately, they do document that at some periods in the summer, they do have those here in Lake Superior.
I used to go to college at Northland. There was like a water study center.
Did they? Yeah. I'll talk a little bit about that. There's been some interesting soap operas about, because a lot of cities rely on tourism and beaches, and so people can be very reluctant to close beaches, and then there's the whole dynamic of You know, it's, yeah, it's funny how money drives a lot of poor decisions when it comes to stuff like this. So believe it or not, that's all the same species of Volvox. But it's a nice colonial picture. So you can't really see the sheath of the gel that's kind of there, but it's kind of like the auger plates that you've worked with in lab. It's kind of like that idea where they're kind of embedded in that. And then it's kind of a hollow center. It's really interesting. You'll see, I'm hoping you'll see these in labs. Sometimes we get sent Volvox that's not this well in the colonial, this large, where you can see it really easily, but sometimes you do see it quite nicely. So hopefully we'll get that. But they're all flagellated. I don't know if you can see the flagella from where you're sitting in this light. The crazy thing is that they all beat their flagella in coordinated patterns so that they can make steady progress from point A to point B. So I don't know how they do that. I'm guessing, I mean, wouldn't they have to do some kind of chemical communication to figure out how to do that? I haven't heard, like, has anyone studied that? I don't know, but you would think they'd have to communicate somewhat in order to do that. And so this is a sexual life cycle going on. That's why you see these bigger congregations here. That's part of their sexual life cycle. But like I said, I'm not getting into that. But in case you're wondering, sometimes it would be easy to assume this is a mixture of different species, but it really isn't. Filamentous, they look definitely filamentous. There's all kinds of different species. Very beautiful. And like, I'm not going to ask you like reticulated chloroplast. You don't need to know that, but that's, you know, they actually get down to that level of detail. Seaweed. The Ulva genus is probably the most clear or example of that morphology. It does really look like lettuce. I don't know if it's edible. Does anyone know? I don't know. Might be.
It probably just tastes like something.
Yeah. So hopefully tomorrow, I think you're going to be tasting some algae if you want. You don't have to. You don't have to, but we usually get a couple different things for you to try because that's often what we're all familiar with when we think about algae. I got in trouble for that years ago because you know, it's for the class. So I didn't pay for it. You know, the class budget fund paid for it. And the dean, why are you ordering food for your class? And I'm like, because I use it to teach about algae. And they're like, what? You know, and so then I explained, can you think of something? I don't really like it, but there's many people who like something that it's wrapped up in algae. Sushi. Yeah. So that'll be one of the things that we'll try. And they are like, why are you feeding your students sushi? What the heck are you doing? And so I had to justify very carefully why I was doing that. And they let me do it. They're like, all right. So I like multimodal learning styles. So, sensory, very good. All right, so this one, there's a lot of debate here. So I want you to be aware that Colocati, that's how I pronounce it. Who knows? Other people probably say it differently. It's a green algae. It's very interesting because it does look very leaf-like. And so they've been looking very carefully at this one. They're like, ooh, those cells look like parenchyma cells, like we see in leaf tissue. They look like some of the cell wall structure is lignin. So that's kind of like what we see too in typical land plants. And so there's a lot of debate, and I put question marks because they don't know yet. But some people say it might be the closest relative to terrestrial plants. But wait, there's more. Now there's this one. So they're saying, well, maybe this one is the closest relative. So Chara, stonewort, which we have around here, they used to be green in the green algae phylum. Then they got their own phylum. So they're in phylum Charophyta. And that's what they look like with the naked eye. They're really interesting plants. We have them around here. And they have at least done enough study on them. They're like, no, they deserve their own phylum. They're different enough from the Chlorophyta. they can, so again, debate. Some, they're like, well, could be the ancestor, the most immediate ancestor to terrestrial plants. Some people are even saying it, they do seem like they're very moss-like. So the saga continues. They're gonna probably change, you'll see new data coming out at some point, and the more they know and discover something else maybe that might. So it's just this debate. So basically, if I ask a question on the exam, what's the closest relative to the terrestrial plants? I probably wouldn't be, like if you said phylum Charophyta or Chara, well, okay. And if you said green algae, well, okay. So it's, and even the Coleochaete. I personally, with what I know, and again, I'm not an algologist, but I tend to favor what they've put up about the Charophyta and the Chlorophyta. I think those are the two most likely, but just be aware there's a lot of debate and don't get too worried about it. All right, green algae, a little bit about them. We certainly have a lot of green algae around here. And that's because we have a lot of fresh water around here, so there's a lot of green algae. And don't forget, they're in lichens. You can see green algae just not even in a lichen relationship on a tree trunk. Like you'd need a hand lens and microscope, but you can isolate them from that. You can see them on the surface of soil, snow. I'll show you some pictures of snow that have algae on it. Rocks, they can just be loose. They don't have to be in a lichen relationship. You've probably seen green sloth fur when they show documentaries of sloths. That's algae hanging out in their fur. Polar bears too, apparently. I've never seen that video of polar bears, but I have a sloth. Sloth, that's a hard word. So I tend to favor this or Charophyta, green algae or Charophyta as a terrestrial plant. So that's what I'm comfortable with right now. Why did they say green algae or Charophyta? So we both have chlorophyll A and B, so do terrestrial plants. They use starch to store extra carbs. Some of them have cellulose, hemicellulose pectin. That's all what we've talked about when we talk about the anatomy of land plants. They even have similar flagella. They have phytochrome pigments. We've talked about all that. Okay, so this is some of the evidence they're using for why green algae. And so just be aware that there's some nice evidence out there for that. All right, we got to stop. We'll continue on with green algae on Friday, and then we'll try to get into bryophytes too if we can.
Right.